Hello everyone. Today we are going to discuss a new topic which is related to medicinal chemistry. And the new topic name is antihistamine agents. Antihistamine drugs are also known as histamine antagonist. Before going to be discussion of the antihistamine drugs, we should know about the histamine and autocoids. So, antihistamine drugs are the drugs that competitively block H1 histamine receptors are conventionally called the antihistamine drugs. Now come to the autocoids. Before going to be discussion of the antihistamine drugs, we should know about the autocoids. Actually, autocoids is a term. Autocoids are substances formed in a various tissues have complex physiological and pathological action and act locally at the site of synthesis. They have a brief action and are destroyed locally, hence they are called local hormones. Actually, they differ from true hormones which are produced by specific cells and reach their target tissues through circulation. The word autocoid is derived from the Greek word that is autos, which means is self, and akos, that means remedy, that means self remedy. Okay, now come to the autocoid. Actually, autocoids are classified into three categories, that is amine, peptides, and phospholipid derived autocoids. Under amines, histamine and 5-hydroxytryptamine are elaborated, and under the peptides we have studied about the angiotensin and kinins. Phospholipid derivative autocoids are containing the prostaglandin, leukotrienes, platelet activating factors. Now come to the histamine. Histamine is also known as tissue amine, that means histose means is tissue. Histamine is a biogenic amine which is formed in many tissues. It is also formed in venoms of bees, wasps, and other sting secretions. Now come to the next point, that is synthesis of the histamine. Histamine, we could say the IUPAC name of histamine is 2-imidazole 4-ethylamine, which is biosynthesized by decarboxylation of basic amino acid histidine and is found in all organs and tissues of the human body. Histidine on the decarboxylation is confirmed the histamine. Now come to the next point, that is storage, distribution and degradation. In human body, large amount of histamine are found in the lungs, skin and intestine. It is stored in the granules of the mast cell and basophils in an inactive form. Actually, degranulation of the mast cells releases histamine, which is quickly degraded by deamination and methylation. Non-mast cell histamine found in brain serves as a neurotransmitter. Next point, the enterochromaffin cells in the fundus of the stomach release histamine, which in turn activates the H2 receptor on the gastric parietal cells to stimulate acid secretion. Now come to the mechanism of action of the histamines. Histamine produce its effect by acting on the histamine receptor. Four subtype H1 to H4 are known. These are all G protein coupled receptors: H1, H2, H3, H4. And the location of the receptors are H1 receptor Location is smooth muscles, endothelium, lungs, blood vessel, gut and brain. Actually, H1 receptors antagonists block the many of the actions of the histamine related to allergy and inflammation. Now come to the S2 receptor, that is stomach, heart muscles, brain. These are present in these gastric glands, mediated gastric gland secretion by the parietal cells. Now come to the next, that is H3, which is presynaptic autoreceptors, which controlled by histamine release from the neurons in the brain. Now next is H4 receptor, which is present on leukocyte, CD4 cells and mast cells have recently been cloned. H4 blocker seems to have therapeutic application in chronic inflammatory conditions. Hope you understand about the mechanism of action, introduction and synthesis of the histamine drugs. Thank you.